All right, well, we're getting started here. Dr. Mwangi is going to be lecturing for the next, I guess, 3 lectures. So he'll lecture today, and then he'll lecture 2 times following Thanksgiving. And hopefully everyone saw that notice that we had to do a little switching. Hopefully that was okay with the presenters, that we switched days on you. If you're having issues with that, let me know, but hopefully that works for everybody. The other thing I wanted to mention is that, so today we'll have Dr. Mwangi lecture on Wednesday. We've got 4 presentations, student presentations: Jason, Allison, Kylie, and April. So look forward to seeing what all you've discovered in your research and your literature research. So look forward to those on Wednesday. And then also there is quiz 4 that's due this week. This is on lectures 14 and 15. Today's lecture is lecture 15, so I'll only quiz on how much material is covered today. So I won't cover things that you haven't heard about today in lecture 15. And so that won't be available until Wednesday, and you have up until Sunday to complete that online quiz on lectures 14 and 15. And of course, I'll be grading your immunoassay assignments. I haven't quite gotten those yet. Any questions on anything? Assignments, the plan ahead. Of course, no class for Thanksgiving, which is next week. So no class next week, so get a break. Any questions, clarifications? Okay, I'm not getting any takers. I guess I'm crystal clear. I can't believe it. I rarely am crystal clear. Okay, well, I'll go ahead and turn it over to Dr. Mwangi, and if you want to introduce yourself and get started, because I know you've got several topics to talk about. Today's on tolerance, the difference between tolerance, autoimmunity, and hypersensitivity. So some really big topics.
Okay.
Thank you, Dr. Mwangi.
Okay, good afternoon, and thank you, Dr. Adams.
You might need to jump in front of the camera and wave at everybody that can't see you. They only can hear you at this point.
Oh, so that was
there he is.
Okay, yeah, so
okay.
By way of introduction, my first name is Waidaka, last name is Mwangi, and it's always a pleasure to teach these 3 lectures, or to offer these 3 lectures. And as you're going to note, it's going to be sort of a high overview or high level overview of these topics, because there's a lot of materials we can cover, but we don't have the time. And by the way, my voice is hoarse. I'm under the weather. I'm taking some antihistamines and so forth. I don't have COVID. I'm COVID negative, but my voice is a bit rusty. So I'm going to take it lightly, but feel free to pause and ask questions as we go along. So we'll be looking at tolerance, or immunity, and hypersensitivity. And when you look at this information, you're going to see that all those 3 are interrelated somehow. And it's going to make a little bit more sense as we go along. So by way of definitions, so what is tolerance? So in brief, or in a layman's language, that would be tolerance is unresponsiveness to an antigen. And that occurs because what happens is that at the very early stages of lymphocyte maturation, that's when the lymphocytes get educated to recognize self versus non-self. So if they get exposed to something at that stage, it doesn't matter whether it's a true self or not, then they get programmed to recognize as self. And you're going to see the implications of that. And once that has happened, that lymphocyte is forever programmed to recognize that as self, okay? Such that even if it gets exposed to the same antigen in future, it does not react to it. On the other hand, autoimmunity is the extreme end whereby you have reactivity against self-antigen. And that is mediated by several mechanisms. And these mechanisms do arise from responses mediated by B cells and T cells. So you have antigen-specific antibodies and antigen-specific cell-mediated immune responses. And these are directed against self-antigens or self tissues. And so under severe conditions, it might end up now precipitating into what is termed as autoimmune disease. And you're going to see and study a few examples of those. So this lecture has got about 60 slides. So if we don't get done today, don't worry. Typically, I spread all my slides throughout the 3 lectures, and we typically end up covering everything within those 3 lectures. So we'll do fine. All right, so how does tolerance arise? Okay, so the presence of TCR or antigen binding receptors, the way they get generated, and I think that's what you went through when you talked about the VDJ recombination and then junctional diversity. During that process of generating the TCR and the BCR repertoire, that process is very random. And in fact, it turns out to be that from 20 to 50% of the TCRs and the BCRs end up recognizing self. So what happens here is that because of that high frequency, there's danger, such that if allowed or if not controlled, you might end up having an individual that is having this very high number of autoreactive BCR, autoreactive TCR. And hence, what happens is that has to be rectified. And the way that occurs is that you have generation of what's termed as self-tolerance, that whereby these self-reactive lymphocytes have to be destroyed through first mechanism called negative selection. And the negative selection occurs during maturation of these immune cells at the primary lymphoid level or the primary lymphoid organs. Now, that primary level of management of these potentially self-reactive lymphocytes, it is not 100% foolproof. Some of those lymphocytes end up leaking out, and so they are taken care of at what is termed as secondary lymphoid organs, and that is they are turned off, and you're going to see some of the mechanisms that do kick in to be able now to take care of that. So let's look at a few examples of tolerance. BVD is a very easy one to talk about. BVD occurs, it's a very bovine viral diarrhea virus. It's a very common disease of cattle. But what happens is that if cows get exposed to BVD at the early stage of pregnancy, and it turns out to be the cow is not immunized, then the virus can make itself to the fetus, and then the fetus gets exposed to the virus very early on during when the lymphocytes undergo maturation. And then the fetal immune system gets trained to recognize the virus as a self-component. And from that point onward, you end up having now that the fetal grows up, and of course, the neonate ends up being seronegative because it does not react to the BVD. And what happens is that for life, or for the rest of its life, it's tolerant to that virus, which is generalized because this so-called persistently infected calf sheds the virus throughout the rest of its life. So, and you can see that it occurs during that window when the cow is pregnant, just that exposure. And then the fact that the virus is so small, it can make itself all the way to the fetus and get exposed to the lymphocyte maturation process. The other example here is this phenomena called dizygotic twins, whereby you have 2 embryos that arise, gives to twins, but their placentas are fused, which means they end up now having shared blood circulation. Bone marrows are shared at that very stage, which means cells from one can make their way to the other. And because of that, they get tolerized to each other. Okay, they are supposed to be 2 independent entities because these are not monozygotic. They don't arise from a split embryo. So what happens is that because of this sharing of the bone marrow, and you end up generating what are termed as chimeric calves, whereby their cells are tolerant to each other, or the immune system are tolerant to each other, such that they accept grafts from each other with no problem at all. And in fact, when you look at cytotoxic T cells from one, they don't react or reject grafts from the other. Okay, so those are two examples of tolerance. So when we switch gears here and talk about the mechanism of induction, we are going first of all to focus on central tolerance, which occurs in primary lymphoid organs, which are the bone marrow, the pia patches, and the thymus, and then the bursa in birds. So what happens here is that during the development of lymphocytes, they end up now reacting to self-antigens, and what happens is that process we talked about of being educated to recognize self, and here specifically now we are looking at T cells. You end up having potentially autoreactive lymphocytes being eliminated by a negative selection. So the thymocytes make their way from the bone marrow to the thymus, and it's within the thymus that that selection process takes place. And as I mentioned, that those now that leak out or escape negative selection within the thymus, within the periphery, they are eliminated. And so which means that the mature lymphocytes that recognize self-antigens in the periphery, they end up now being removed through several mechanisms that we are going to talk about. And some examples here are given, are indicated here, whereby T cells within the periphery, one of the mechanisms that they can get tolerance is exposure to low antigen doses, especially exposure to those antigens in the absence of co-stimulations. And that scenario leads toward a stem-based energy. And you also have now induction of apoptosis, and also regulatory T cells come into the scene to be able now to mediate that process. And I'll walk you through that in a short while. So let's get started with the cellular tolerance. So how does this happen? So once you have immature lymphocytes being generated in the bone marrow, they have to go to the thymus, where is their kindergarten of training. They have to get trained there. But the thymus is only a very limited representative of the organs within our body. How then does the body make sure that the lymphocytes are exposed to all the antigens that are present in that body or in that individual? You have this transcription factor called AIR, or autoimmune regulator, which is a unique machinery that promotes expression of self-proteins that are actually made by different organs within the body. So, such as insulin, thyroglobulin, or myelin-impressive protein. So, in other words, the thymus, if you consider that to be like Congress, the representatives there are presenting the issues from each state. So the AIR would be this mechanism that is actually bringing all the issues from the states, and they are all present within the thymus, such that the thymocytes that are going through that process, they are going through the thymus, they are exposed and educated to recognize this itself. Okay? So, but there are exemptions. The exemptions are such, there are certain exemptions which include certain antigens that are secreted in certain specialized organs or specialized tissues or specialized parts of our body, such as the brain, the eyes, and the testes. And those, they get protected because there's mechanical barriers that are actually secluded. So I think you've gone through the process of negative selection, but here, I want to just walk you briefly, whereby the pre-T cells, when they enter the thymus, if they react too strongly, they bind it too tightly, they end up, you have induction of apoptosis, then they get deleted through negative selection. But there are those that bind mildly, they bind mildly. So here is actually telling you that you have no binding, but the truth here is that a thymocyte must bind to a self-MHC, either MHC class 1 or class 2, in the context of self-peptides. And if it binds very, very moderate, in a very moderate way, then it gets selected. That is not through the positive, that is what is termed a positive selection. If it fails to bind at all, it gets ignored because it does not receive any signals. And so it gets eliminated because it does not get the essential signals that it requires from an antigen to present itself within the thymus. So that is now what the mechanism by which now you end up now having generation of this pool of thymocytes or T cells that end up now populating secondary lymphoid organs. Okay, so how does this process sometimes fail? This process might end up failing when you have defects in the air, that autoimmune regulator gene, such that you don't have proper presentation of these other antigens that the lymphocytes are going to get exposed later on in life. And so failure, or when you have no defects in air, that can lead into autoimmune diseases, because now you end up having generation of lymphocytes or T cells that react to self-antigens. On the other hand, you also have, if you have defects in this gene called FAS, FAS is actually required because, or CD95 is required because this is the receptor that is involved in apoptosis. Majority of those 20 to 50% potentially reactive lymphocytes that we talked about, they are eliminated through apoptosis. And that requires the role of the CD95. So if it's not functional, then you can imagine that those defective or those potentially autoreactive T cells are actually going to survive. So then they don't undergo clearance. Okay. So switching gears and talking about now mechanism of central tolerance with regard now to B cells. B cells, you know that they are generated within the bone marrow, and then they undergo differentiation within the bone marrow. So within the bone marrow, B cells get exposed to self-antigens, and again, if there's very high affinity binding to self-antigens, two things can happen. One, you can have non-negative selection through induction or apoptosis, so that potentially autoreactive B cell gets eliminated. But unlike T cells, B cells have got one more unique capability. They're able now to sort of take a back step through what is called receptor editing. They switch genes around and reconstitute that BCR, such that that BCR now becomes has less binding to cells. So its potential to end up being autoreactive gets eliminated. So that recombination allows a significant pool of these B cells to be salvaged, so that now they end up now populating secondary lymphoid organs. So if we may look at some of the events that occurs within the bone marrow with regard to B cells, is that you have now the mature B cell, and of course exposure to low doses of antigen can be one of the mechanisms by which now you can have deletion of B cells, because too low of an antigen, the BCR ignores that, especially short polypeptide or highly soluble proteins are ignored by B cells, especially in the absence of costimulatory molecule. So this information that I'm presenting here is in a nutshell is actually telling you that the BCR is very, very selective, or it's sort of sensitive to what it gets exposed to, which means that low doses, the B cells are unresponsive. But what happens is that you require moderate doses of antigens, especially complex antigens, for there to be antibody production. But on the other hand, if you're having too much or very high doses of an antigen, you can induce what is called activation-induced cell death. Okay, so let's look at events that do play a role in eliminating potentially reactive T cells within the periphery. So as mature self-reactive T cells, leave the thymus, they can be, if they leak out and escape into central lymphoid organs, there are three mechanisms by which they can get eliminated. One of those is energy, which is basically functional inactivation. And this happens in the scenario whereby you're having these T cells recognizing self-antigens, but there is lack of adequate levels of costimulatory, which is very critical. The second mechanism is deletion. And this deletion occurs in a rather unique way, a mechanism called activation-induced apoptosis. So in this scenario here, which is rather unique, as I said, is that you have these potentially autoreactive T cells upregulating fast and fast ligand to the extent whereby fast ligand from one can actually bind to the fast receptor on the other. So you have this population of T cells that end up killing themselves through upregulation of both fast and fast ligand. The third mechanism is suppression by Tregs, or generation of T regulatory cells, which end up producing these immunosuppressive molecules such as TGF-beta or transforming growth factor beta and IL-10. So let's look at these examples in a little bit more detail. So under normal circumstance, when you have an antigen-presenting cell interacting with a naive T cell, you have three signals. You have TCR recognizing a peptide in the context of MHC molecules. Then you have costimulatory molecules. And of course, there's that critical need of the CD28 interacting with the B7 complex. Those three critical signals are required for the APC to be able now to activate a naive T cells. And once the threshold has been met, you have the activated T cells undergoing proliferation and secreting cytokines and so forth. This scenario here, in a majority of cases, requires that you have to have a source of inflammatory reactions or inflammatory signals. And some of those end up now coming directly from pathogen recognition receptors, or you have inflammation that is being provided by cytokines and chemokines. Otherwise, if you lack that, you don't have a productive activation of T cells. In the case of energy, you have a scenario whereby the naive T cell has the capability to recognize a peptide in the context of MHC molecules, but it lacks the costimulatory molecules. And that's very, very critical. And once that naive T cell gets engaged and receives that signal at that very age, it is programmed to become allergic the rest of its life, such that if on subsequent interaction with a highly activated APC, it's incapable of getting activated. Concurrent to that, you can also have engagement of an antigen-presenting cell, whereby an APC or a T cell recognizes a peptide presented in the context of MHC, but instead of now provision of costimulatory molecules, you have provision of inhibitory molecules. And one of those is CTLA-4, which CTLA-4 is cytotoxic T cell lymphocyte associated antigen 4. This molecule here gets upregulated when a T cell gets activated beyond a certain threshold, such that it switches from making CD28 and it starts now making CTLA-4. It turns out to be that CTLA-4 has got a much higher affinity for the B7 complex than CD28. And that is actually a homeostatic mechanism to be able now to switch from that activation step to be able now to modulate that response. Otherwise, that lymphocyte is going to undergo, or you might end up now having that cell dying because of poor activation. So CTLA-4 is presented now to a naive, if you're having a T cell engaging, if you're having a naive T cell recognizing a peptide, but it is receiving this inhibitory receptor or signaling by the CTLA-4, it becomes allergic. The other molecule within this category is called PDL, or the programmed cell death receptor. And again, these are inhibitory molecules that end up now inducing energy. Okay, so the other example I wanted to give you here is suppression by Tregs or T regulator cells. And again, the first part here is actually showing you a productive activation of naive T cells by an APC that requires or that entails recognition of a peptide by the MHC, and then you have provision of co-stimulation and inflammation by cytokines, and then you have a productive activation of T cells at the end of differentiating, and they're able now becoming functional. But on the other hand, you can have a situation whereby you have suppressive T cells asserting their role in a much better way, such that once you have Tregs being generated, they can actually destabilize the events that I've described. The events that I've described here in two ways. One, the Tregs can engage this activated APC and switch the activation signals off, such that the APC is no longer capable of activating a naive T cell. That is one way that the Tregs can do it. But the best way they do it is actually by secreting immunosuppressive cytokines, such as TGF-beta and interleukin-10, and they're able now to dampen T cell activation. So I wanted to pause there and find out whether there is a question so far before I switch now to talking about B cells. So with regard to B cells within the periphery, there are several mechanisms that those B cells that escape being removed within the central lymphoid organs, they can get eliminated via several mechanisms. So under normal circumstances, what happens is that mature B cells populate secondary lymphoid organs, they get activated, they end up now producing plasma cells that end up making antibodies. But if this B cell potentially has capability of being autoreactive, it can end up being eliminated by several mechanisms. One of those is through what is termed as exhaustive antigen challenge. And this exhaustive antigen challenge, I need to clarify here, is not confined to potentially autoreactive B cells. What happens here is that mature B cells, if they get exposed to too much antigen stimulation, they can undergo what is clonal exhaustion, and they end up losing it. That's now when you're formulating a vaccine. You have to be very careful that you don't overdo it, because the B cells you want to make or to induce to make antibodies that you require, you might end up killing them. Okay. The other mechanism is that within the periphery, you have B cells recognize an antigen, but in the absence of co-stimulation. So then they become allergic. Or you could also have exposure to an antigen in the presence of excessive suppressor T cell activity. And that also, again, has an effect on the outcome of the B cell activation. The last one here is actually quite common, especially when the B cells get exposed to antigens that have got repeat epitopes in what is now termed as excessive T cell independent antigen, such that now the B cell lacks CD4 T cell help, because the BCRs within a B cell can get engaged by multiple epitopes in these antigens that has got these repeat epitopes. Those antigens examples include carbohydrates, nucleic acid, because they have those repeat motifs. So taken together, all these can end up resulting in what is called functional deletion of T cell subset. Okay. Now, the last one here of excessive T cell independence can also result into what is termed as receptor blockade, whereby because of the presence of this antigen that has got multiple repeats, you have engagement of these multiple BCR that end up now secluding other receptors to get engaged, and thereby the B cell cannot get help, but cannot be able now to interact through or signal or get signaling by other receptors because of this receptor blockade. Now, there are also other mechanisms that do play a role in peripheral B cell tolerance. And one of the next example I wanted to talk to you about is what is called idiotypic networks. So in simple terms, idiotypic networks is an antibody against another antibody, in very simple terms. And how does that arise? The way this works is that if this is an antigen and you have a defined epitope, you're going to have a BCR recognized in that epitope, and they end up now producing a paratope. That paratope has got what? The complementarity determining regions. And you remember that the affinity maturation occurs here by mutations, and you have now unique amino acids that arise over here. But those amino acids are not germinally recorded. So which means this paratope, once it has been generated, is sort of like a new antigen that the immune system has never gotten exposed to before. So because of that, you can have another B cell now recognizing those unique motifs and making antibodies against those. So this antibody 2 can actually now neutralize antibody 1, or B cell number 2 can actually be able now to have a BCR or secretional antibodies that blocks B cell number 1. So idiotypic networks, that's how they arise. Okay, and so on and so forth. And they kind of, because now every time now you have generation of a new paratope, okay, you potentially generate a new antigen, and so it can go on and on and on. That's why it's called a network. Okay, so the last one here is actually quite interesting because it's something that we deal with on a constant basis. And this is peripheral B cell tolerance mechanism, whereby presence of pre-existing antibodies do moderate or do control B cell response. How do we know this? So neonates that have got very high maternal antibodies are very poor responders if you immunize them against that antigen. And why is that the case? This is the mechanism that is at play. So this data here is actually showing you antibody titers, and then here you're showing weeks post, time in weeks, and it's shown here that if you immunize a neonate in the absence of maternal antibody, it responds very well very early on. But if you do that to a neonate that has got very high levels of maternal antibodies against the antigen, you don't induce an immune response, and that's why they recommend wait for X number of months before you immunize. So exactly what's happening here, it is because of this mechanism that involves this receptor called CD32 or Fc gamma receptor 2B. This receptor here has got very low affinities for IgGs, which is good because so long as there are low level of IgGs, it does not kick in. The threshold has to be high enough for it to be sensitive or for it to be involved. And so what happens is that if you're having very high levels of antigen-specific antibodies, IgGs in the blood, those IgGs will interact with the antigen that you are trying to, that you're injecting in there. And what happens is that this complex here now, or the binding of the IgGs to the antigen, allows the Fc region of these IgGs to open up, and the CD32 recognizes that. That antigen, remember that it has multiple epitopes, so you end up now having a concurrent recognition of that antigen by the BCR as well as the circulating antibodies. Under this condition, you have this bridging effect whereby the signaling by the BCR is actually weaker than the signaling by the CD32. The CD32 signaling under this scenario is much more powerful. So it ends up now shutting off the signaling by the BCR, such that that B cell is inhibited from responding. And that's how now you end up inducing energy, or you end up now modulating B cell responses in the presence of these high levels of pre-existing IgGs. Okay? So, with that, any questions?
Yeah, any questions or clarification? That was a lot of new information. Some overlap from what we've studied before as far as central tolerance, but the peripheral tolerance is somewhat new. Are there any questions, clarifications?
Okay, if now let's go to autoimmunity, and again, we try to be having a high-level overview of this topic here. And autoimmunity, as we alluded to, mentioned before, is a condition that entails or involves antigen-specific antibodies and or concurrent involvement of cell-mediated immune response against the body's own tissues. And the outcome here is that you end up having clinical lesions or clinical symptoms that are as a result of hypersensitivity reactions. So autoimmunity can occur because of multiple factors. So one of those is genetic predisposition, and you're going to see some examples of those. But in a number of situations, you have involvement of hormonal influence, or that the hormones do drive autoimmunity. So in general, you might end up having normal immune responses, but these immune responses end up being directed against previously hidden antigens, which means that the lymphocytes, they didn't have a clue that these antigens existed because they never got pre-exposed to them. And those previously hidden antigens, it could be that they are arising because of tissue damage. That these antigens were there, but they were secreted. They were less cytosolic antigens, they are not exposed. Okay, but under situation of less necrosis, all of a sudden an antigen that was secreted is now exposed. But you could also have generation of new antigens through molecular or structural alterations, and there are several mechanisms that do play a role here, and you're going to study those. Last but not least for this one is that as an individual grows or develops, you have synthesis of new antigens, and those new antigens, the lymphocytes were not pre-exposed to those. On the flip side, you can also have normal immune responses, but because of a mechanism called molecular mimicry, you can end up having an immune response directed against microbial targets, but those immune responses are cross-reactive. You're going to see quite a number of examples with that regard. On the flip side, you can also have abnormal immune responses because of failure of regulatory controls. For example, the ones that we talked about, failure of apoptosis. Or you could also have virus infections that end up now destabilizing immune responses or triggering unusual immune response. And you're going to see some example here, especially with our canine distemper virus, that it is uniquely known to drive this process. And the last example here is this mechanism called microchimerism. What is microchimerism? This is when you have leakage of cells from a fetus into the mother. And if it turns out to be the fetus is male, the fetal cells with the Y chromosome end up in the mother, and they end up establishing what is termed as microchimerism. And so you end up having certain mothers that are actually Y chromosome positive because of microchimerism. And because of expression of Y chromosome and encoded antigens, it can end up now resulting into induction immune response that could fall under this category of autoimmune reactions. Okay, so let's look at some examples of this. Abnormal immune responses to normal self-antigens. And so genetic predisposition is one of those. And the example is what we looked at, mutations within the air, said that now it does not function properly as expected, which means that there's going to be a gap in the thymus such that the lymphocytes don't get exposed to or get educated to certain host proteins. The other example here is failure of apoptosis, whereby you have failure of potentially reactive lymphocytes failing to get deleted, so they end up thriving. The last one here is MHC genes and predisposition to autoimmunity, which is quite common. And this table here summarizes or gives a summary of a few of these. One of those that has got very high correlation is ankylosing spondylitis. This is now the inflammation of what? The spinal cord. You've seen folks who end up having this rigid spinal cord that they cannot be able to walk straight, because this is has been shown to be associated with this MHC class 1 MHC molecule. This is B27, and the frequency is quite high. It's greater than 95%. Okay, narcolepsy. What is narcolepsy? When you are always, have someone who is always very sleepy, even during the daytime, it is associated with this MHC class 2 DQ6. Okay, now just picking another one, is insulin-dependent diabetes mellitus. You can see how you have now 2 MHC molecules that are involved. So on. You're going to see some of these examples later. Now you can see that these MHC genes predispose those individuals to autoimmune reactions, because they end up now favoring, or they end up having a unique setting whereby you have that individual having a certain immune outcome that ends up resulting in some of these reactions that we're talking about here. Okay, so besides on MHC, the other condition here is the other example I wanted to give here is with regard to initiation of autoimmunity, is this scenario whereby you have normal immune responses to abnormal or unusual antigens. And here is the example that I had given earlier on, whereby you have exposure of lymphocyte, mature lymphocytes to hidden antigens, and they end up now, of course, reacting to those, and these fall under several categories, and they could also arise because of several reasons. One of those is, as cells age, the structure also changes, and they get certain domains or certain components getting exposed, or you have cells being damaged, and you also have a release of previously protected or hidden antigen. The other one here is molecular alterations, and this occurs in antigens, and as mediated by several factors. And the best one here is what is termed as rheumatoid factor. I'll walk you through this shortly. And this is driven by an infectious agent. The last one here is molecular mimicry, whereby immune responses directed against a pathogen can end up now cross-reacting with a self epitope. And we'll see a few examples here. Okay, so just one more time here now, just to go over these hidden antigens again, is that these neuronal T cells, once they meet these previously hidden antigens, they are able to react in a manner that is unusual, in the sense that they are going to require certain scenarios to actually be functional. In the sense that if you don't have enough information there, or provisional, let's say, co-stimulation, it might not amount to anything. But remember that, for example, if you take humans, you're always getting inflammation because of, let's say, infections and so forth and so forth. So there's that idea of what you termed as bystander effect. Okay, so it has to be a microenvironment that ends up now being optimal to allow these non-trained T cells to react to these molecules that spill off, let's say, from the eye or from the spermatozoa, or you have now newly synthesized antigen, let's say, as we undergo puberty. Okay, so the last point here included, because this happens quite often, tissue damage. And tissue damage results into these squalid antigens. When somebody is having a heart attack, one of the easiest things they can look for within the blood is what? Molecule called cytochrome C. Cytochrome C is released from where? Mitochondria. When somebody is having a heavy heart attack, you end up having a lot of cytochrome C being released, and they're able now to use that as to measure actually and tell you, oh, this is what's happening. And so release of these sequestered antigen can also triggering an autoimmune reaction. One more example here is, and this is something that happens because the turnover of red blood cells is quite high. As red blood cells age, the structure also changes. And so this molecule called band 3, which is a critical, band 3 protein is critical for transport of proteins for ions, transport protein on ions in the RBC. This protein is hidden and masked by a lot of carbohydrates. But what happens as the RBC ages, it starts falling apart and it gets exposed. So what happens here is that you have exposure of new epitopes that are recognized by autoantibodies. And those antibodies end up now oxidizing these RBCs, and the RBCs are removed by phagocytosis, and they end up now destroyed, taken up, and destroyed by phagocytes within the spleen. Wanted to change gears here and talk about a unique example here of antigens generated by molecular alterations by talking about this so-called rheumatoid factor, because you see these a lot, especially in dogs. So what happens here is that canine distemper virus can induce IgG responses, but quite a number of these antibodies, they bind to these antigens or antigens in the canine distemper in such a way that structurally, the structure of the IgG, specifically within the FC region, gets exposed. So typically IgGs, the FC region is hidden until the antibody binds its target. Now, in this case here, the binding of the IgGs to these canine distemper antigens is such that unique FC epitopes are exposed, and what happens here is that IgMs, preferably IgMs that derive from the B1b, remember the so-called natural antibodies, natural IgMs, they end up now binding to these new FC epitopes on the IgGs that are specific to the canine distemper. And this results into formation of a complex. And given the pentameric structure of the IgM, it can be able to bind up to what? Up to 10 of these IgGs. And if these IgGs then are binding to the canine distemper antigen, right there you have an immune complex. And what happens is that these rheumatoid factors end up depositing in joints, whereby now they end up causing inflammation and inflammation, activation of macrophages, activation of other other molecular release of cytokines that results into damage of the cartilage because of the inflammation that is induced through those mechanisms. The end point here is that you end up having high levels of TNF alpha that end up now causing cartilage and bone marrow and bone destruction. So rheumatoid arthritis is a big deal, especially to the elderly. And of course, management of TNF alpha using TNF alpha neutralizing agents is a mechanism of mitigating, is one way of actually managing rheumatoid arthritis. So with regard now to molecular mimicry, this is quite simple in the sense that you have induction of immune response against an epitope derived from a pathogen. And it turns out to be that epitope is shared with a host cell or host epitope. And there are several examples. One of those is Trypanosoma cruzi. This parasite here expresses an antigen that is very similar to neurons and cardiac muscle antigens that leads into autoantibodies that are specific or end up binding to central nervous system and also bind to the heart and mediate autoimmune reaction. The other example here is antibodies directed against group A streptococci, M antigen, specific epitope within this M protein. Once the host has made antibodies against that, it turns out to be this M antigen has an epitope that is very, very similar to an epitope that is present on cardiac myosin. So those antibodies are able now to bind to the heart tissue and mediate tissue destruction. Poliovirus, VP2 antigen. Antibodies against this antigen, they also can bind acetylcholine. And once they bind the acetylcholine receptor, they are able to block the signaling, and that causes myasthenia gravis. Last but not least is multiple sclerosis, which is a condition that arises when you have Epstein-Barr DNA polymerase specific CTLs directed against epitopes on this antigen. And those CTLs recognize similar epitopes on myelin basic protein, whereby now those CTLs end up now causing demyelination, and that end up now having this negative outcome of multiple sclerosis. Okay, so how do you manage all this? One, you can end up using immunosuppressive drugs such as glucocorticoids to also production of cytokines and inhibit inflammation. And the other one is use of drugs that can actually inhibit breakdown of arachidonic acid. And some of those, some of these drugs include prednisone and dexamethasone. The other drug of choice here is azathioprine, which is an inhibitor of nucleic acid synthesis. And this is a very good blocker of highly proliferative T cells, especially CD4 T lymphocytes. Cyclosporin A is another blocker of T cells, but this one works by blocking IL-2 synthesis. So with that, we've talked about those two topics of mutants and autoimmunity, and I wanted to pause there and ask for questions before we move on into hypersensitivity reactions.
Any questions on these different mechanisms of and triggering the immune response to react to ourselves? There's lots of really interesting mechanisms, and it's always interesting to me that pathogens would want to even mimic what the immune response would see. It seems like it's counterintuitive that you would develop an antibody response to a pathogen that will also attack the body. I don't know how that's an advantage for the pathogen.
It could also be a random event. Yeah, it could also be a very random event, you know.
Random,
because when you look at it, that because when you try to look for the advantage, sometimes you can't be able to connect the dots, you know. So you start wondering, you know, and some of these shared epitopes, you know, the mimicry there is not perfect, but the TCR, the BCR is able actually to cross-recognize them.
Yep.
Yeah.
Any other comments, questions? All right. Well, I told everyone online that if they think of questions, you can send them to me via email or send them direct to Dr. Mwangi if you have questions that he can answer again when we have them in class on the 27th.
Okay. So why don't we talk about hypersensitivities? And again, here we are talking of 4 different hypersensitivities. Typically, when we offer these in at least 4 different lectures. So we are squeezing these all 4 into part of this lecture. So let's see how much we are going to cover. And again, you know, pardon my hosiness. So what is hypersensitivity reaction or intolerance? These are undesirable reactions produced by normal immune system, including allergies and autoimmunity. And these reactions could be uncomfortable, damaging, and occasionally they end up being fatal. Okay? Take home message here. You need to know that for there to be a hypersensitive reaction, you must have had a pre-exposure. In other words, hypersensitivity reactions is a recall response, is a memory recall response. And in a majority of cases, the first time you get exposed, you did not even have a clue that you got exposed because you feel nothing. You feel nothing at all. So these hypersensitivity reactions are classified into type 1, 2, 3, and 4. And we are going to go through this one by one and look at some of the core components or the most important components and how they are involved in these reactions. So type 1. Type 1 is the most common and the one that is well known. And the major effector molecule is IgE. So you need to know that IgE is the driver of type 1 hypersensitivity, and it occurs within minutes, only if you have had prior exposure. And once that system is engaged, there are major cellular effectors or cellular players. And number one are the mast cells. Number two are eosinophils, and then you also have basophils being involved. What are the outcomes? Allergic reactions, asthmas, anaphylaxis. Okay, those are the critical syndromes that are associated with type 1 hypersensitivity. Type 2 involves the role of IgGs and IgM. And it occurs within hours if there's prior sensitization. But the cool thing here is that, or the most important thing you need to know here is that these IgG and IgM, they mediate their effector function by engaging complement activation. So the complement here is the driver, is actually the one that is doing the heavy lifting here. And what are the outcomes? Cytolysis, okay? Antibody-mediated cytolysis. And of course, we're going to see some examples here whereby if it's directed against RBCs, you're going to have anemia, thrombocytopenia, neutropenia, and so forth. Type 3 involves IgAs to the lesser extent, but mostly IgGs. But although it occurs within hours, just like type 2, here you need to differentiate between type 2 and type 3 because in type 3, the neutrophils are the ones that are now driving that process. Remember in type 2, it is what? Complement. In type 3, you require neutrophils to be engaged, to be activated. And once they're activated, once they're on the scene, they end up now mediating their function because through the molecules that they release. The molecules that they release are the ones that end up now causing damage. And you're going to see examples of that. The last one here is type 4 hypersensitivity. And this is termed as cellular mediated hypersensitivity reaction because it involves the role of activated Th1, activated CTLs, and activated macrophages. And since it occurs several days after the fact, after exposure, it is called delayed type hypersensitivity. And the outcome from there is formation of granulomas, cellular proptosis or cell death, mediated by these effectors. Okay, so let's look at type 1 hypersensitivity and talk a little bit about how it works and the role of mast cells and IgE. So you might, let me maybe remind you a little bit here because it's very possible you've covered this. IgE occurs in our blood at the lowest amount. It occurs at microgram quantities in blood, and it has the shortest half-life. But although that is the case, it doesn't mean that it's harmless or that it's incapable of mediating its function. You're going to see that, yes, it does work very well in terms of asserting and be able to drive its biological functions. So let's look overall at the events that happens when you have involvement of IgEs in the case of this type 1 hypersensitivity. So the scenario is such that for you to get IgE, you have to have B cells that have been polarized to Th2. And there are specific conditions that allow production of IgE because the inducing target or the inducing molecule is specific. And in this case, you're going to find that allergens are actually very good at driving that. And why are they so good at that? Allergens happens to be one of those antigens that have got those repeat units, which means that they're able to bind to a B cell and activate a B cells in the absence of help. But because of lack of CD4 help, you end up having not only insufficient or poor costimulatory, but you end up having the antigen driving B cell responses to a Th2 and production of IgE. Now, in the case of Th1 immune response, remember, you're only going to have an allergic reaction if you have had pre-exposure. Meaning, the initial time that you reacted to an allergen, you produce some level of memory, so you have some circulating antibodies, IgE in this case, against a certain allergen. Unlike other receptors, the receptor for IgE can allow the IgE to sit on that receptor in the absence of the allergen. In other words, for IgG and other receptors for antibodies, the immunoglobulin receptor is able to bind once the antibody has bound to its target. But in this case, if you're having very low levels of IgE, they can sit on the IgE receptor. And that's exactly what makes type 1 hypersensitivity very risky. Because the only thing you need, and in minute amounts, is the allergen. Because as soon as it sits in, and because of the fact that it has these repeat units, it's able now to ligate all these receptors, and boom, the mast cells undergoes degranulation. So you don't need a lot of the allergen to be able to trigger that process. So let's see what happens here. Before we talk, we go through step by step. Just wanted to look a little bit about these effector cells. We talked about mast cells. They are mostly within the skin, the connective tissue, and mucosal surface. And they are loaded with these vasoactive substances, mostly histamine, heparin, and in ruminants, you have serotonin, okay? And the cells, they express the high affinity receptor, the Fc epsilon receptor. This receptor has got the highest affinity for any immunoglobulin, and so which means that you need minute amount of IgE to trigger signaling via these receptors. So basophils are similar to mast cells, but these are only found in almost found in blood. And eosinophils, also present in blood. These ones are endowed with multiple effector proteins or effector molecules, including major basic protein, neurotoxins, which endows them with the ability now to kill helminths and other parasites. We'll come across that later. And in addition to that, they are phagocytic, and they are able now to mediate their killing through antibody-dependent cell cytotoxicity for various destruction. We are going to see that when we study mucosal immune response. So let's look at the sequence of events if you have a prior sensitization to a certain allergen. So what happens is that once you have now re-exposure to an allergen, and you have ligation of multiple receptors on the mast cell, within minutes, actually within seconds, you have the mast cell degranulation and you have massive release of histamine, depending on the quantity of this allergen. You have this immediate release of histamine, serotonin, enzymes such as tryptase, proteases, and proteoglycans. And it is these molecules that end up now starting asserting their biological function and causing damage at the local site of release. But within minutes, you also have release of these lipid byproducts, the echinocoid synthesis and secretion of leukotrienes. And leukotrienes are very good at not only being inflammatory molecules, but also they're chemoattractant for other leukocytes, such as neutrophils and so forth. Now, you also have release of prostaglandins, platelets activating factor. Now, prostaglandins, how do you know that prostaglandins are being released. Prostaglandins, when they bind to the receptor, that's how you start feeling pain. Okay? That's how you start feeling pain. So, but that's not the end of the story. That's not the end of the story. What happened is that so long as that allergen is sitting on those receptors, within 6 to 24 hours, you have this massive upregulation of cytokines, quite a number of them, IL-4, IL-5, 6, and so forth, TNF-alpha, and some chemokines. Now, we can't study all these. I just want to pinpoint one of them because it is a major player in this scenario, and that is IL-5. IL-5 is a potent attractant of eosinophils, but that's not the end of it. It does not only attract them, but it also activates them. And once they are activated, eosinophils end up releasing the major basic protein. And these molecules released from the activated eosinophils are involved in epithelial cell damage. So taken together, you end up having these activated eosinophils end up now exacerbating or making the situation worse. Making the situation or the damage caused the mast cells is actually worsened by now the recruitment and activation of neutrophils. Okay, so, but are the mast cells intending to release to, why the immune system is actually not deploying type 1 hypersensitivity with all those negative outcomes in mind? No, the intent of type 1 hypersensitivity is actually to eliminate the allergen or self-eliminate roundworms for helminths, in the gut. And that's the sole purpose of that. But a lot of these other molecules that end up being released and causing damage, you can call that as bystander effect. Okay, you have casualties that arise because of all these side effects that are caused by the release of these molecules, such as now you have mucus formation, runny nose, and so forth, because of that. But again, these molecules are needed in mediating the type 1 hypersensitivity desired biological function. Okay, so end of the game is that what you need to understand, or the take-home message here, is that two purposes of the type 1 hypersensitivity is to eliminate the allergen or cure the intruder. In this case, the roundworms or helminths that are invading, and once it's able to achieve that, then you can be able now to appreciate the fact that that is the intended goal, and everything else that is happening is a side effect. Okay, so I wanted to look at a few examples of type 1 hypersensitivity, and this actually is split into two. First one is localized allergic reactions. Those happen within a very localized tissue within the host. And this example here is allergic rhinitis or hay fever. And this is manifested by increased mucus secretion, inflammation of the upper airways, sinuses. Another example here is allergic inhaled dermatitis, and this is very common in Dalmatians. And you can see some example of the reactions that is observed in these dogs. You also have bronchial asthma, lower airways inflammation, and that's quite common in cats. And the associated hyperactivity, inflammation, and tissue injury manifests there very, very clearly. Eosinophilic granuloma complex that is observed or has been documented in dogs and cats, and this is mediated by now the role of those activated eosinophils, and you can see that it turns out into this kind of ugly outcomes because of that type 1 hypersensitivity reaction. You also have parasite allergies in pets, and that's quite common. You also have food allergies, okay, and those have been well documented, whereby you have the outcomes being quite manifested in and demonstrated in various ways. And one of those, you end up having increased peristalsis due to contraction of the intestinal muscle and so forth. So those are examples of localized allergic reactions. But you can also have what are termed as systemic allergic responses, which might also end up being life-threatening. Okay, and this example here would be vascular permeability that might end up leading to massive edema. And the example given here is you have epicloral swelling, and if it compresses the trachea, you might end up now obstruct and impede breathing. You can also have systemic reaction that results in fluid accumulation in shock organs. Okay, shock organ here being that the default organ in which that host end up now having massive reaction. For example, in cats, that's going to be the lungs. In dogs, that's going to be the liver. And so what happened here is that in under this scenario here, you might have a situation whereby you're having a massive blood and fluid loss that might result in drop in blood pressure or decrease in cardiac output. You can also have constriction of the bronchioles leading to asphyxiation. Okay, so in dogs, fluid loss from the gut and visceral pooling of blood does or can occur in liver. So how do you treat this? Well, epinephrine is something that might be very common to you. And those who have hypersensitivity reaction, you've heard of folks now who carry EpiPens. Okay, EpiPen is especially when it's just a self-injectable epinephrine that is a lifesaver. Okay, the other treatment is use of chromonine sodium. That this is an inhibitor of intracellular calcium increase, because during those events that you're talking about within these responsive cells, the mast cells, and the neutrophils, or the mast cells, intracellular signaling involves calcium signaling pathways and chromonine inhibitor. This chromonine inhibits that intracellular signaling pathway and ends up now mitigating that outcome. The most common one here is use of antihistamines. Okay, and basically these ones here end up now blocking the effect of histamines by this. Some of these are actually inhibitors of just the histamine receptors. Under serious conditions, of course, if somebody has an allergic reaction, they have an EpiPen, and and you can judge that the reaction is the host or the individual is not really responding very well. You rush the person to the hospital. They end up now administering corticosteroids, and the way those corticosteroid drugs do work is they end up now stabilizing the membrane so that now you have reduced or inhibition or breakdown of the membrane components, so that now you inhibit production of arachidonic acid and thereby inhibit release of prostaglandins and other inflammatory molecules, and be able now to reverse the process. One example here that I wanted to talk about here that has been shown to work is called preventative hyposensitization therapy. And in this case here is the basic approach is use of the allergen in certain measured doses or in certain formulations to drive the immune response to switch from making an IgE to make a different isotype, especially IgG. And in this case here of this dog here that was allergic to the ragweed, what happened here is that they show that by increasing exposure or immunizing this animal to with the increasing doses of this ragweed, they were able now to stimulate it to switch from making IgE and start making IgGs against this ragweed pollen, the ragweed pollen, and whereby the allergic reactions were solved through this hyposensitization therapy. You can also do the same thing by, let's say, taking that allergen, formulating that with a cytokine that is going out to skew from a Th2 to a Th1 and achieve the same goal. Or other option is induction of or stimulating Tregs that would now dampen the Th2 that favors IgE production. Okay, so any questions there regarding now type 1 hypersensitivity?
So there was a question here online, would vaccine reactions be associated with type 1?
It depends on the inducing component, because certain individuals have ended up having a type 1 hypersensitivity to certain stabilizers in the vaccines. Yeah.
Oh, that they're responding to the stabilizers in the vaccines?
Yes, like BSA and so forth. And when you go now to approval process, let's say the World Health Organization, before they realize that the use of albumin or BSA was for vaccines for human and dogs was very, very high, and then they end up finding out it was inducing this hypersensitivity reaction and so forth. And so those dosage levels, the recommendation was actually to either eliminate it completely or reduce those. Because if you're going to be using those vaccines that are formulated with those stabilizers, remember the first dose, nothing happens. But the moment now you come and boost that individual, if they're reactive to that component, that's now when the allergy kicks in.
So with this desensitization therapy, so does it induce all three of these mechanisms? Blocking antibody, biasing the immune response to Th1, and induced tolerance? Is it known which of the three is actually working?
No. It's actually depending on formation, it is going to be driving towards one path. For example, in this case of this patient here, they were able to show that they were able to stimulate switching, isotype switching of the B cells for them to stop making IgE, and then they start making IgGs. And the maths was actually very simple. What you need is more B cells making IgGs than the B cells that are making IgEs, because if you're having a dominance of IgGs compared to IgE, every time you have that allergen being exposed, this dog being exposed to that allergen, the first antibody they are likely to come across is going to be what? IgG as opposed to IgE. And which means then you're able now to solve that problem. So each one of these mechanisms, yes, the approach here is that it's either or. You have to really maybe work hard enough to really drive multiple differentiation patterns. But again, you don't need to. If you can be able not to successfully be able not to switch one case, this scenario here to one of these others, for example, switching from Th2 to a Th1. And of course, that's going to be favoring what? IgGs, maybe IgAs and so forth, so that you end up solving that problem. Because induction of tolerance for especially allergens, that's a little bit tough. Okay, but if you come up with a very strong pro-inflammatory molecule, you can actually switch into a Th1 much more easily than induction of tolerance.
Great, thank you. Any other questions besides mine? I don't see any other questions. Is this a good stopping point, or should we continue?
It's a good stopping point. And then we can pick up from here. We have several slides. We'll finish those in the next lecture. And again, my apologies for my hosiness. Hope to be in better shape next time.
You sound very good. I didn't notice anything as far as
some antihistamine.
Oh, you're on histamines. Speaking of allergies.
Yeah. So it took some painkillers. Yeah. So, but again, that's fine. Yeah.
Okay.
So we can pick up from here and talk about type 2 and move on from there. We'll have type 2, type 3, type 4, then we cover the rest of the topics. Yeah. All
right. So we'll cover that when we see each other on the 27th.
Yes. Have a happy Thanksgiving.
Yes, you too. You too. And otherwise, I'll see everybody else on Wednesday for student presentations. Thank you so much. Have a good night.
Good night.
Good night.
Good night.